Bismillahir Rahmanir Rahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to see another lecture that is related to the hematology, and it is hematology or oncology, that is about the fine needle aspiration cytology. So in the previous videos lecture, if you guys remember, we talked about the bone marrow aspiration, in which we talked different cancer cells, and we talked the bone marrow, what is bone marrow biopsy, each and everything in detail. Now fine needle aspiration cytology, or we can say that fine needle biopsy, both are almost similar to each other, but in case of the aspiration we take the fluid, and in case of the biopsy we take the tissue portion. Okay, although both are the same, in both of the cases we mostly study the cancer cells. So in this video we are going to see that what is fine needle aspiration cytology and how we can perform it, procedure, staining purpose, each and everything. So let's start the video. Now in this video we are going to explain the introduction, that what is fine needle biopsy. We are going to see the purpose, we are going to see the requirements, site for fine needle aspiration, procedure, staining, examination, and what we can see in fine needle aspiration biopsy. So first of all, let's start the lecture with the introduction. So fine needle aspiration cytology, this is again the diagnostic procedure in which we mostly evaluate or examine the lumpus or masses, means the extra outgrowth in the body. Okay, when we examine it we call that fine needle aspiration. Now in this case what we can do, we can insert the needle at that particular area where there is a lumps or masses or extra growth, if there is or swelling if we see. So in this case we inject the needle or insert the needle and will take the fluid or tissue, for example we can say cells, and then we will make a smear on the cell, will place it on the cell, will fix the cell, and then examine the slide under the microscope. Now in this fine needle aspiration, at the first stage of the fine needle aspiration we will examine the benign and malignant cells. Benign and malignant cells, that is the natural presence that is mostly present in this case. Okay, benign is the normal cells, and we can say non-cancer cells, and malignant is the cancer cells and can spread throughout the body. So it is invasive nature, relatively quick results. Now let's see the purpose, why we perform this fine needle aspiration. Again in this case we mostly do that process to just examine and determine that the lumps, or we can say that the masses or the outgrowth that is present in on the body. So we can see that either this is cancerous, this is caused by the cancer cell, or non-cancer cells, means it is the benign or malignant. So we can see this condition through fine needle aspiration. Moreover, through this technique we can also give the appropriate treatment to the patient. For example, if we see that there is a malignancy, so then we mostly recommend surgery or chemotherapy or radiotherapy. What type of the treatment the patient needs, so we will provide that appropriate treatment to the patient. Let's see the requirement, that what we required in case of fine needle aspiration. So in this case we required PPEs, personal protective equipments, like goggles, gloves, cap, overall, mask. Each and everything is too much important while performing that technique. Another thing is that we need a fine gauge needle, and it is 22 to 25 gauge. Okay, this is really important. We need syringe. Syringe is important for the aspiration, means through which we can take the liquid portion. We need microscope, staining techniques, slides, local anesthesia. In this case mostly the anesthesia is not given, because very less pain is happen, or the patient can feel less pain. but still we can provide the anesthesia in a very rare cases. Okay, this technique is also important when there is abnormal when we see the abnormal report in when there is abnormal blood report, so then we mostly recommend the fine needle aspiration test. Now these are the site as you can see. I have just put only 6 pictures, but there are also some other sites, but these are the most common site through which we can perform the fine needle aspiration or biopsy. Mostly it is can perform by this thyroid area and this breast area. But if you see the outgrowth or lumps, or we can say masses at any particular area, so then we perform the fine needle aspiration biopsy. Like in this case, there is a lumps at the wrist portion, there is also the lumps in the finger, there is a lumps at the shoulder area, so masses are available at the neck portion. So this is the thyroid swelling, this is another so these are the specific points and these are the areas through which we can perform fine needle aspiration. At that particular area, we can sometimes we take the fluid if it is filled with fluid, or sometimes we can take the tissue or the any part of the body that is important to check whether the body is affected by the cancer cells or the non-cancer cells. So that's why it's important to see. These are the most important sites. Now let's see the procedure, that how the whole process can be performed. First of all, we have to place the position, the exact position we have to place the patient. Now we have to locate the area where there is outgrowth or where there is a masses or lumps is present. So we have to locate that specific area and will place the patient according to that position. After that, once we know the location, each and everything, so then we will wash our hand and then we will put the PPEs, personal protective equipments. After that, we will locate the mass portion and will palpate it. Once the mass portion is located and we palpate it, so then we will sterilize that particular area or the skin, and then we will use the aspirate needle and will inject it into the at the lumps area or at the masses area, and then we will move it back and forth. Due to the back and forth movement, what will happen, the cells or the fluid will be easily taken, or we can easily collect the cells. After that, we will put the cells or the specimen into the we will put this into the fluid or onto the slide, or we can make the smear, we can say like that. And once the smear is made, so then it should be fixed using alcohol, or we can then place it at a room temperature, then it will be dry. After that, we will use different staining techniques like hematoxylin stain, eosine stain, Quick stain, Pepenculi stain. So these are all the staining techniques. Then we will use all these staining techniques. Now after the staining technique, we will examine the slide under the microscope. Now this is the simple diagram, and this is showing that how you can do the fine needle aspiration. So this is the scanner area, this is the scan. So first of all, we have to use the scan, and we will using the scanner area, and then we will once the affected portion is detected by the scanner, so then we will inject the needle, as you can see. Now once the needle is injected, so then we will take the fluid. Okay, so this is how the whole process is performed. This is another diagram that can showing the fine needle aspiration procedure. So in this first picture, as you can see, the sample is taken by this needle, and then it is placed onto the slide. After that, then we can make the smear. Let's see the fixatives material that we are using in fine needle aspiration. So in this case, we use ether alcohol 95%, ethanol 80%, propanol, pure methanol, or 10% formalin. You can use any one, Okay, so remember, the main thing is that whenever you are making the slides for the fine needle aspiration, so you have to make 10 to 15 slides. That's really important. You have to make 10 to 15 or 20 slides sometime we can make. And some of the slide you will fix it with ether alcohol. Some of the slide you will fix it with 95% ethanol, 80% propanol, or pure methanol, and 10% formalin. So you can you have to fix all the slide with this particular some of the slide you will fix it with alcohol, some of the slide will ethanol, some of the propanol, some of the slide methanol, and some of the slide with formalin. Now, what are the purpose and what are the principle of this fixative solution? It is important to prevent the cell from the shrinkage. It will kill the microbes. Moreover, it can enhance the staining. That's why it's really important. Let's see the staining technique that we can use in the fine needle aspiration biopsy. So in this case, we can use Pepenicillio stain, Romanesque stain, Giemsa stain, special stain. In special stain, we use Alcine, Persian blue, Congo red, Mason Fonta, and then we use Zn stain. We use He stain, Graham stain. So all these are the staining that we can use. It means that some of the slide we will stain it with the Romanesque, some of the slide we will stain it with the Giemsa, and so on. Okay, so it doesn't mean that we can use, okay, as we make the 15 or 20 slides, so some of the slide we will stain with Romanesque, some of the slide with Giemsa, and so on. So we use all these stain. Now under the microscope, these are the different pictures that I have taken. So under the microscope, if you wanted to see the cancer cells, so these are the cancer cells under the microscope. Now this is the Romanesque stain, and if you see this field, so this is actually the normal salivary gland tissue that are showing grape-like cluster of epithelial cells composed of the spherical, acine, and branching ducts. Okay, this is the first field, and this is the salivary gland area. Now this is another field that is done by this Pepenicillio stain. In this case, you can see benign acer cells that are arranged in the Rosti formation, and the cell have abundant foamy vacuole cytoplasm with indistinct cell border and acosine nuclei. This is another picture. As you can see, in this case you can see that the slide is stained with the Romanesque, and it can showing short tubular segments of ductal epithelium composed of small hypochromic cell characteristics of the chronic cellulitis. Okay, so this is another portion. This is another the field, and this is also Romanesque stain. And this is the spirate of benign lymphoepithelial cysts are characterized by the mixed population of lymphoid cells dispersed in watery background. This is another. Now these two are the cancer cells, and these are the, we can say, these are the most important field through which we can identify the cancers. This first picture is actually the cellular polymorphic adenoma, and it's showing the discrete and cluster of epithelial cells, and it is done by He stain, hematoxylin stain. Then the second picture is showing the long strand of spindle cells embedded into connective tissue stroma. This is also He stain. Now these are the other fields of the microscope. This first picture is actually the Pepenicillio stain, and it can showing the cluster of epithelial myoepithelial cells associated with the fragment of myxoid and chondroid stroma are characteristics of polymorphic adenomas. This is also the cancers. Okay, now the second is the Romanesque stain, and in this case these are the myxoid chondroid stroma of the polymorphic adenomas, often has a this fibrillary character along its edge. The third picture is also the cancer field, and in this case these are the myoepithelial cell component of the polymorphic adenomas, frequently has plasmocytes appearance. These two are another fields, and the first picture, this is the field which is stained by the again the Pepa stain, and in this case these are the and mixed population of the large flat sheet of oncocyte and lymphoid cells. The epithelial cells have abundant dense eosinophilic cytoplasm, well defined cytoplasmic border, broader enlarged centrally placed nuclei and prominent nuclei. The next picture is showing aspirate Wertherine tumor are characterized by a mixed population of lymphocytes and clusters of the epithelial cells with abundant granule cytoplasm. The cells lies within a dirty proteinaceous background. So this is also the Romanowski stain. Then we have DQ stain. So in DQ stain, when we see this field, so this field is actually showing the tightly cohesive cluster of the basaloid cells and many background strip nuclei. And this is remember guys, the field which I am showing here, this is actually the cancers, and some of them are the benign, some of them are the cancer. But this, as I already told you, that you know, this test should be done, or the technique should be done when there is abnormality. So that's why it's really important. The next slides, or the next portion, showing the membranous variant of basal cell adenoma. There is a dense hyaline extracellular material surrounding the basaloid cell cluster. This is the next picture that is showing predominantly discrete oncocytic cell in the fine care aspiration smear. This is the MGG staining technique, and this is again the Romanowski stain, in which we use oncocytomas that contain epithelial cells with abundant granular cytoplasm identical to those seen in Wertherine tumors, but oncotomas lack the lymphoid components. This is another field, and in this field we are using the hyaline variant of myoepithelioma, showing dissociated plasmocytes cells with abundant cytoplasm and excentric nucleus. Then we have another field, that is the small round cells with scanty cytoplasm along with pinkish hyaline globes, and this is MMG stain. Now this is another field that is also the cell arranged around the pinkish globes. So this is the pinkish globes, and these are the cell around it. It is also showing the cancer. Okay, now these are also the cancer cells, as you can see, and it is the adenoid cystic carcinoma, and this is actually the neoplastic cell having oval to round hypochromatic nuclei with phenyl to corsal granule chromatin and scant cytoplasm. They are associated with hyaline globes which stain pale grey green in this preparation. These three are the field, as you can see, and these are also the cancer field. And this first field is showing the acine cell carcinoma that are characterized cytological by irregular cluster and sheets of epithelial cells with foamy granule cytoplasm and bland nuclei. So this Romanowski stain. Next we have the some cells of acine cells carcinoma, and it contains the characteristics red granule within their cytoplasm. So this is the Romanowski. The third one is also the Romanowski, and in this case we can see acine cell carcinoma having finely nucleated foamy cytoplasm. These two are another cancer cells, and this is actually showing the abundant dissociated squamous cells with nuclear enlargement and polymorphism. Occasionally mucus secreting cells are also seen in this case. As you can see, mucus are also secreted. And then we have another field that are showing high grade mucoepidermoid carcinoma. The cells are large, polymorphic, and show severe cytologic atopia. The cytoplasm have dense quality with squamous features. Then we have another paper stain which are showing high grade salivary duct carcinoma, and there are flat sheets of epithelial cells with abundant delicate cytoplasm and moderate nuclear atypia. The nuclei are round to oval in shape and show finely granular chromatin and prominent nuclei. Abundant nucleosis is associated with the malignant cells. Then we have another cell, that is the tight cluster with scanty to moderate cytoplasm. So this is H stain. These are another cells, and in this case these are the aspirate form. Hemagonomes have a bloody background in which are scattered a small number of cluster spindle cells. These cluster may have tubular appearance. As you can see, these are tubular appearance. The nuclei are bland and elongated. So this is the Romanowski. Then we have again hematoxylin and eosin stain, H stain. So in this case we have this is actually the lymphomas are characterized cytological by tissue fragments with a lacy-like appearance. The individual cells have abundant clearly cytoplasm and small dark nuclei. As you can see, small dark nuclei is in this case is present. Then we have again pepper stain, in which the aspirate from granular cell tumor are characterized by the sensitile groups of cells. So these are the sensitile group of the cell with abundant granular cytoplasm surround bland round two oval nuclei. The background of the smear has a dirty granule appearance. As you can see, this is a dirty granule appearance. Then we have hematoxylin, and this is the amyloblastoma. This is the amyloblastoma, and these are the tightly cluster of basaloid cells along with loose connective tissue stroma. This is the Romanowski stain, and it is the aspirate form schwannoma, contain tissue aggregate with indistinct cytoplasmic membrane. Schwannomas contain spindle cells. These are the spindle cells, as you can see, with filamentous cytoplasm. Nuclei are blunt or point with nuclear bands. Then we have DQ stain that have well differentiate keratinized squamous cells. Carcinoma smear are characterized by high cellularity with abundant enucleated and nucleated keratinized squamous cells. This is the DQ stain. Poorly differentiated squamous cells carcinoma smears show cohesive and discohesive group of cells with vascular nuclei, coarsely granule chromatin and large prominent nuclei. No evidence of squamous keratinization is seen in the specimen. So this is also the pepper stain, as you can see. The next slides is showing is the next two field. So in the first field, this is the pepper stain, and it can showing melanomas are characterized by discursive spindle of epiploid cells with prominent nuclei. So this is the pepper stain. Melanin pigment may be seen in the melanoma. So this is a melanoma, okay, and this is about the H stain. Then we have Kaposia sarcoma, contain the radial arrangement of the elongated spindle cell. This is the Romanowski stain. So these are all the fields. There are also some field, but these are the most common field of the cancer cells identification. When you there are also some similarity, okay, there are also some similarity with the normal cells, so it required a very high skill and expertise to identify the cancer cells. But these are the most common fields, as I already told you, and it's mostly seen in the case of the cancers. Now let's see the advantages and disadvantages of the fine needle aspiration. So this technique is very rapid. It have a very minimum invasive, and there is no need of anesthesia in some cases, in most of the cases, and it is very specific to diagnose the malignancy, and it can give us this accurate, it is very accurate and simple technique. Disadvantages is that it required high skill and need a high expertise, and sometimes it may cause septicemia and hemorrhage. Now contraindication means in which situation the sample should be not taken: when there is hypertension, when there is hemorrhage, when there is bleeding disorder, when there is bacteremia, septicemia, and when there is pancreatitis. At that situation the sample should be not taken. So this is all about the bone marrow aspiration. So thank you so much for watching. I hope you like this video. If you like this video, so make sure to hit the like button, share this video, subscribe the channel, and also press the bell icon to get more interesting videos like that. Thank you so much for watching.